what I wanted to do today was just give you a bit of an update, give you a sense of where we were. And again, this is not all the data we've collected for the Bali project, but just a select number of examples. And for those of you who are in our team whose data I've not drawn on, it's not to say I'm not interested or I don't think it's significant, but I was trying to put together in 15 minutes as coherent a talk as I could. So there are a couple of omissions that don't reflect necessarily on how good your data might be or how interesting it is. So just to give you a quick summary, I've tried to boil down or simplify some of our research questions a little bit. We framed it slightly differently in our proposal. But in this case, the simplified form, I suppose, is this notion that we were trying to understand in our project the impacts of land use on biogeochemical processes, trying to understand in tandem or in parallel what those impacts were on functional diversity, and then trying to understand those linkages. How could land use be then impacting functional composition? What are the knock-on effects for biogeochemical cycling? Disentangling to some extent the effects of functional diversity versus direct sort of land use effects. And so, in that respect, that's going to be the main focus of what I talk about today and trying to, shed light on that. So, again, we've got more data than what I'm showing, but just a subset here. And I'll sort of zoom through a couple of bits that you may have seen before. So one of these bits of data that I need to highlight, because I think it's really important, is some of the data that Terry's been collecting, and you're probably well aware of it. A lot of work that Terry and Yadvinder's group have been doing looking at carbon dynamics across the study site. I think in terms of the so just some nice images, nice pictures of the different components of flux that are measured. And again, sort of taking some of these data from Terry's paper, sort of the headline result being high levels of productivity across the land use gradient. No sort of significant difference between total MPP amongst logged versus old growth systems, but differences in allocation, differences in the relative components. So old growth forests showing higher degree of canopy production, so leaf litter production and so forth, whereas logged forests showing higher rates of woody growth. And in terms of understanding net ecosystem carbon balance, I think there's an implication, if we tie into some of Terry's work on soil respiration and heterotrophic respiration overall, that the net ecosystem carbon balance of these log systems might be significantly depressed compared to the old growth systems, because we're seeing a greater proportion of sort of soil and woody debris carbon stocks being turned over and released back to the atmosphere in those log systems. And as a result, if you do sort of the net ecosystem carbon balance sort of calculation, sort of back of the envelope, you see quite a substantial reduction in what would be the net amount of carbon storage. Some of the work that we've been doing on the girdling experiment has provided some insights into how root systems are contributing to turnover of organic material and heterotrophic respiration and so forth. These are some data. I apologize for not sticking the name on it, but these are data from Andy Nottingham, who is doing work using automated lichor chambers, sampling around a number of different trees, different taxonomic groups and so forth. And I think these are some of the plots, some of the time series he collected for a number of different plots around specific trees. And what he found that was quite interesting was really that different individual trees linked to their species identities show different trends in respiration rates after girdling. So in some cases, those plots over the top show quite a sort of a relatively linear decline. These plots at the bottom, it's a bit noisier. I mean, in each of the cases, you see a significant reduction in overall soil respiration after you girdle, and that's linked to sort of shutting down root activity. But the actual trend over time varies. So this, broadly speaking, suggests that this linkage between the sort of plant-soil linkage, at least in terms of the respiration component, is partly influenced by sort of species identity and something about sort of the individual sort of physiological properties of those trees that he sampled. And this is a sort of a bit more of this data, and I think what was quite interesting here is he was looking at, again, sort of time series using these automated lichor chambers, and what he was finding is that there are sort of distinct sort of diurnal trends that vary before and after girdling, right? So before they girdled, you saw a large diurnal fluctuation where the daytime sort of respiration rates were enhanced compared to nighttime rates. And that makes sense if you're thinking that plants are assimilating carbon, in their canopies, some proportion of that carbon is going below ground to roots and emerging as, as respired carbon. What he found after girdling was there's sort of a transition period after just under 2 months, 50 days, where you see the reversal of that nighttime respiration exceeding daytime respiration. It's not entirely clear what generates that, why it flips. I think it's easier to kind of mechanistically kind of understand pre-girdling why you have a higher diurnal respiration rate, because that's probably linked to sort of canopy root linkages in terms of photosynthate. Okay. But again, some interesting trends emerging, that imply how root activity, rhizosphere activity might be then linked to overall sort of soil system processes as I said earlier, potential evidence of an imprint, I suppose, of species or taxonomic group identity. As you're all probably aware, and I've talked about this before, I think we've done a lot of work on understanding functional trait distribution. So as trying to understand sort of functional diversity across the landscape, we implemented a number of different sampling campaigns across the gradient, looking at plant functional traits, looking at the characterizing the microbial community composition using molecular techniques, as well as doing some surveys of invertebrates and so forth. And I think one of the largest single chunks of activity was this work on plant functional traits, especially foliar traits that Sabina and David Burslam were involved in, where they sampled all the carbon plots in addition to two in old growth systems in Malaya Basin and the newly established Danum carbon plot. And again, you're probably all quite familiar with this, so I'll try and keep this moving. But I think the highlight point from this is that the functional trait composition of the plant community was really determined by a mixture of direct land use effects from logging, as well as the underlying soil properties. So there are a number of traits, for instance, linked to a nutrient composition that were reflected, so underlying soil properties that were linked to those traits were more strongly affected by underlying soil properties, so those are listed over the top, and a number of other traits that seem particularly pertaining to photosynthesis that appear to be more strongly influenced by land use change, and I suppose in that case it's because selective logging has removed dipterocarps primarily and damaged, any other surrounding trees. So you've selected for a kind of vegetation community that's quite different from old growth. Okay, and so this is sort of summarizing it. I think broadly speaking, what we're seeing is in terms of photosynthetic traits, photosynthetic related traits in log forests, you see a shift towards over traits that are more acquisitive. In terms of plant economic spectrum terms. And some evidence that, and this plays into all the microclimate data that we have, that microclimatic conditions are significantly different in these logged environments. It's sort of internally consistent with other data we've collected. The 13C composition, for instance, of the leaves tends to imply hotter, drier conditions based on the shift in the isotopic values. I'm slightly embarrassed to show these data because the person who generated these data are here, so correct me if I make any mistakes. But we've tried to scale up some of the trait measures using some of the remote sensing efforts. And I think what was really interesting from this exercise is that it highlights to some extent issues to do with scale, right? Because if you take a look at this image of sort of our ground-based trait measurements, there's something that's inherently contradictory, because we're showing a number of traits that are more strongly influenced by land use change. But what was really interesting about Tom's data set when you scale up to the landscape is you actually find that the underlying soil properties or land surface properties linked to topography, soil fertility, and so forth are a stronger determinant of some of the foliar characteristics, like remote foliar N, foliar P, specific leaf area, and all that. They're more strongly determined by elevation and the underlying soil properties, whereas actual land, and so that's sort of these plots shown over the top where you have these sort of continuous distributions of foliar values plotted against elevation. And if you try and sort of map on top of that some of the different individual plots where data were collected from the ground-based measurements, we see that land use effects don't map on as well to that the overriding effect at this bigger sort of landscape scale is really to do with underlying soil properties. And so that's showing to some extent the fact that one of the limitations of our ground-based sampling. Even though that ground-based sampling might be useful for training up or parameterizing your remote sensing data set, once you scale this up with a sort of bazillion data points that you get from something like LiDAR or hyperspectral measurements, you tend to find that the trends, the underlying statistical relationships that you may have found from your more limited ground-based sampling don't hold up at a larger spatial scale because you're somewhat biased in your sampling to some extent, or you've just got a more limited sample set. Microbial functional traits, again, this was work done by the Lancaster team, Dav, Sam Robinson et al., and a couple of folks at Aberdeen, like Furt. And really, they were using a number of sort of microbial characterization techniques, molecular and lipid biomarker techniques. And the headline thing that I'd like to talk about, and this links into some of the trends we see in heterotrophic respiration, is that the shift from old growth to logged forest is reflected in the soil microbial community composition. Focusing down on the fungal communities, we see a shift from old growth systems that are dominated by ectomycorrhizal or have a larger abundance of ectomycorrhizal fungi and a lower proportion of saprotrophic fungi. You see a shift towards one where in the log systems shown in black bars here, where there's significant reduction in ectomycorrhizae. And that, again, is linked to removal of dipterocarps because they are ectomycorrhizal, removing the host plants, I suppose, the symbionts hosts, is having a major knock-on effect. And interestingly, and this sort of chimes or is sort of in line with what we're seeing with some of the ecosystem loss, carbon flux data, we see an increase in the abundance of saprotrophic fungi. We also see evidence of some sort of species-specific effects that could be roughly aligned with a sort of plant economic spectrum. So these are data from Furt, and we have a comparison between Macaranga and Paraschirea, so pioneer versus late successional species. Interestingly enough, and this is sort of data from before and after girdling. So green is before girdling, white is after girdling. And I mean, in terms of soil respiration, he was sampling a smaller subset of taxa. So he didn't pick up as strong an evidence of sort of a taxonomic effect as Andy did. But what he did find for his sample set was that there was significant change in nitrous oxide flux, right? So that's implying that something's changing with nitrogen dynamics when you girdle. For the old growth trees, interestingly enough, no significant reduction in nitrous oxide flux before and after girdling, indicating that whatever sort of rhizosphere processes that are going on, it's not really having any kind of downstream impact on at least nitrous oxide production. In contrast, for the Macaranga system, you see a significant reduction in nitrous oxide flux. I mean, it's not, and this, so keep in mind this is a log scale, so the differences are actually bigger than, in the actual numbers, absolute numbers are bigger than what we're seeing, the differences are larger than what you see here. But you see a significant reduction in nitrous oxide flux, suggesting that there's something about the rhizospheric processes that are enhancing nitrous oxide flux within sort of the rhizosphere of these early successional plants. So an interesting sort of link potentially to functional type, understanding some of the nitrogen dynamics and gas exchange. I'm going to zip through some of the work on manipulations, especially some of the termite work because Rob's already spoken about it. But we also did a number of different manipulations tweaking key parts of the system. One of these was a reciprocal transplant of litter to try and get an understanding of the relative importance of site-specific versus substrate effects. And we also did this sort of exclusion experiment for termites and ants in Malleau Basin where the invertebrate team poisoned basically 1 hectare patches excluding ants or termites. And I won't speak about the invertebrate experiments too much because Rob's already discussed that. But in terms of the leaf litter study, what was really interesting in terms of headlining findings is really that there was no strong evidence of a litter quality effect. It was really a site effect. When we did these reciprocal transplants, rates of litter decay were more strongly predicted by where you put the litter as opposed to where it came from. And that's interesting because the chemical composition, the physical characteristics of the litter from old growth versus log forest are quite different. There's different CN ratios, different nutrient content, and so forth. Generally speaking, litter under old growth conditions was decaying more rapidly. And we think that this might be because in log forest, again, microclimate, it's more extremes of temperature, more drying, evaporative drying, and so forth because of edge effects. That could be inhibiting the rate of litter decay. Interestingly enough, one of the other findings that we think was really quite sort of curious was that in the log forest we saw much more rapid utilization of phosphorus within that litter. And that's sort of consistent with this idea that you might have a replacement of the plant community with trees that show more acquisitive traits. They're going to be accessing phosphorus for growth or recolonization at a much more rapid rate than you would see in the old growth system. So more rapid depletion of phosphorus in litter. Again, so I'm not going to cover this because Rob already did. Some interesting results with our termite suppression experiment in terms of evidence of buffering of soil moisture content, buffering of the presence of termites, buffering rates of organic matter turnover, and so forth. Another interesting, on the ant side, I think what was quite interesting is and this probably comes as no surprise to those of you who know about invertebrates. Unlike myself, I'm a bit of a novice, I think, is that a large proportion of the organic matter resources were being turned over, moved around, and so forth, consumed by ants. The suppression of those ants led to, due probably to a reduction in predation and so forth, led to an significant increase in a whole series of other taxonomic groups. So it'll be interesting, especially as we link and discuss more with Yadvinder about sort of food web studies, for instance, about how to sort of try and figure out what the underlying predictive relationships might be. I mean, what we did here was sort of a relatively simple in the sense of, not easy to implement, but simple in terms of concept. We're looking at a reduction in abundance versus higher abundance. But what we probably need to go to in the future is understanding density-dependent relationships, right? So if you can suppress or reduce populations to known amounts, but across a continuous space, what that translates to, right? As opposed to a presence or absence, but something more nuanced in terms of understanding how differences in abundance or activity of termites or ants might influence sort of downstream processes. We've done a bit of modeling and data assimilation, and again, there may be those of you in the audience who know much more about this than I do. We did try at first to use quite a complex sort of multi-layer canopy model because we have all this LiDAR data, which is Matt Williams' SPA model, but it turned out that wasn't working very well, and I won't go into the details of that because I don't fully understand the issues myself. But what we've fallen back on is using a much simpler carbon dynamics model that he has called Dalek, and David Milodowski, his postdoc, was using the carbon plot data combined with various other observations, like satellite-based observations. Just take a look at the figures on this side, in what's described as a data assimilation scheme, to try and make predictions about how some of these carbon cycle processes are changing over time, such as GPP, NPP. What's shown here on the top, the black dots are I think, from the hemispherical photos, and I think the green is the reconstructed LAI from satellite observations. And what they've tried to do in a data assimilation scheme, as far as I understand it, is use two sources, multiple sources of data and information, and converge on a most likely scenario. And I think the main thing I wanted to point out about this, and I'm almost there at the end, is that what we see in the temporal dynamics are what I think is one of the really interesting one of the interesting features of this data. These drops in LAI, which then correspond to reductions in MPP and so forth, correspond to annual reductions in rainfall. Okay, and what's interesting for the ENSO period is we don't see as large a drop in LAI or productivity as we anticipated, and it wasn't perhaps as didn't translate into as large reductions compared to previous years. So it'd be interesting with these sort of modeling data assimilation approaches to try and probe the temporal dynamics of these systems and try and understand the relationships with climate. So miraculously, my last slide, just in time, or maybe running over massively. But a couple of things. I think there are these mixture of effects. Logging can select for a plant community that has more acquisitive traits. That translates into rapid rates of regrowth, but no net increase in carbon sequestration because things like heterotrophic respiration might be hugely enhanced. We see some evidence of kind of what I would think of as top-down effects, changes to the plant community then affecting microbial community composition, for instance. So reductions in mycorrhizae, increases in saprotrophic abundance, changes in the way that phosphorus is utilized in the litter layer, changes in respiration rates, changes in nitrous oxide flux, all linked to shifts in the plant community mediated by logging. Also, some evidence of bottom-up effects. So I'm thinking here of sort of our ecosystem-friendly, small little ecosystem engineers, the ants and termites that might be buffering against change or in some way mediating changes in resource availability in the understory, which could then subsequently affect carbon turnover. We don't understand this link as well. I think we really need to establish that linkage between the invertebrate community and other sort of soil processes, and I'm hoping that in future we can sort of begin to flesh that out in greater detail. And I'll stop here because I've probably overrun my time. Thanks.